Contents, Volume 1, Forward, Prefatory Note, Acknowledgements, Prologue. Drugs described in Volume 1, Ajacarna, Viteria indica lin, Amragandi heredra, Curcuma amata roxb, Anantamula, Hemidesmus indicus lin, Schult, Aragvada, Cassia fistula lin, Araluca, Ailanthus officinale roxb, Artraca, Zingiber officinale vosk, Aruna, Terminalia aruna roxb, WT, and Arn, Terminalia alata hainax roth, Arca, Calotropis gigantea lin, RBR, Calotropis persera air, RBR, Ashokai, Soraka asoka rox, D wild, Polyalvia longifolia benth, Anthook, Babula, Acacia nilotica lin, Del, SSP, Idica benthbrennan, Acacia leucofloa rox, Wild, Acacia farnesiana lin, Wild, Bacula, Mimiosops elingi lin, Balataka, Semicarpus anacardium lin, Chacromarta, Cassia obtusifolia lin, Chinaka, Caesaretinum lin, Corica, Angelica glauca edu, Ganti, Baliospermum montanum wild, Mule, Arg, Dava, Anagisis latifolia DC wall, Ex bed, Dravanti, Jatropha circus lin, Ella, Eletaria cardamomum lin, Matan, Amumum sebulatum rox, Amumum aromaticum rox, Amumum capulaga sprague and Burkle. Further, the use of ultraviolet light has been helpful in analytical and FAR methods have been introduced for the identification and evaluation of the drug Pharmacognostical work. Gradually, the trend of study has changed and the modern trend is therefore the broad, scientific understanding of drugs based on biology, chemistry, and coordination with collateral sciences which are the fundamental basis of pharmacognosy. Various methods and procedures have become a source of menace to public health. Besides identification of the drugs been devised by pharmacognosists from time to time, as and when, adulteration by macro and microscopical methods, fluorescence analysis, detection of the active constituents present in the drug is also equally important. It has been the concern of pharmacognosists to explore new vegetable drugs, ascertain their identity, source, and make a systematic botanical and chemical examinations to study the active constituents. Each drug has its specific method of evaluation. Some require organoleptic, some microscopic, some physical methods. Well, for a number of drugs, chemical methods are applied. Study of the market samples, along with the pharmacognosy of authentic specimen. The investigations the commercial samples of the same drug should follow, without which the study remains incomplete, because the majority of those available in the market are either heavily adulterated or entirely replaced, sometimes by cheap and worthless substitutes dressed up to resemble the genuine article. Many drugs are sold under the same name, and sometimes the same drug is sold under various names in different parts of the country. There is a high proportion of spurious drugs and also genuine drugs of unacceptable quality in the market. First step is a complete survey of the drug in different drug markets and procuring samples sold under the same name from different parts of the country. The market sample should be compared with the genuine one by the methods described above. Often more than one method is employed to detect adulterations. Adulteration due to admixture of exhausted drugs are usually ascertained by chemical methods aided partly by organoleptic and or partly by microscopic methods. So far, the study of market samples. The classical methods of analysis in pharmacognosy and botany are applied, and the samples are evaluated against the following criteria to determine their acceptability or otherwise. 1. Source. Collection from the plant considered to be the genuine source. 2. Part. The correct part of plant, like root or bark or whole plant, as reputedly used. 3. Appearance. Form of supplied samples. Size. Health as judged by color, odor, texture, and taste on the basis of organoleptic tests. 4. Filth. In the form of molds, insects which preclude the acceptability of drugs. 5. Extraneous matter. Percentage of substances like sand, earth, straw, grass, other plant parts. Parts of the same plant, but not known to contain active ingredients, etc. Up to 10% was kept arbitrarily as admissible. but over 10% was treated as unacceptable. Pharmacognosy in India, a brief historical account. Its development and growth of Pharmacognostical School. Although it is only in the very recent years that pharmacognosy has substantiated its claim to rank as a science, yet the collection of herbs for medicinal use has remotest antiquity. Ayurvedic treatises and medical lexicographs mention detailed descriptive characteristics of vegetable drugs, their geographical distribution and habitat, soil suitable for their growth, the season for collecting them, the duration of their efficacy and the methods of their storage, elaborate classification of drugs and medicinal plants and detailed instructions on every conceivable point, such as the gathering time for different types of drugs, parts to be collected, have been given. It will be interesting to note that the nomenclature in case of some drugs and synonyms used provide clues for drugs' identity, source, geographical distribution, and its action also. The perusal of literature of Ayurveda from the time of Charak and Sushruta shows a peak of systematic development. It made its way far beyond the shores of India, into Egypt, Greece, and Rome. Dioscorides mentions in his work about a number of aromatic group of drugs, for which India has always been famous. Indian perfumes and other things were in great demand in Rome and other countries preceded. 1949, for instance, Ctesias of Knidos, who was physician to Artaxerxes Neman about 400 BC, refers to cinnamon oil exported from India. 1939, there is evidence to show that an extensive trade in Indian drugs existed between India and Rome many centuries ago. Pliny also refers to a heavy drain of Roman gold to India in buying costly drugs and aromatic spices. Pliny, Historia Naturalis Volume 24 pl. In the 8th or 9th century AD, Charak's work was translated and was well known in Arabia. Raises 865 to 925 AD calls him Scarak. Avicenna 80 to 1035 AD quotes him as Sirach. And Serapion mentions Charak by the name of Zark. The fact that several standard Hindu works on medicine and materia medica were translated into Arabic and that several Indian drugs like pepper, lac, nard, licorice, asafoetida, osmum sanctum, bdellium, cinnamon, myrrh, red sandal, calamus, acorus calamus, and the chebulic myrobalans have been included show the extent the Arabic medicine was influenced by the Hindu medicine. Ray, 1902, Prasad, 1949, Locke, Sit. During the invasion of India by the Greeks, Scythians, Huns, and the was mutilated or lost medicine, however, had maintained its progress during the Buddhistic period, and a large number of vegetable drugs were added to the Mohammedans successively. A good deal of the existing Ayurvedic literature already extensive materia medica. By the 11th century or so decadent that in generally in knowledge, learning, and practice of, both in medicine and surgery, there is evidence to show that even in the early Buddhistic period, gardens were established for growing drugs and herbs. When the Mohammedan rule was established in India from the 13th century onwards, the Arabian or the medieval Greco-Arab system, also known as the Ananitib, became the state system of medicine. The Arabic system brought with it a rich store of its own materia medica, and during the long period of Muslim rule up to the 17th century, this system came in contact with the Hindu system of medicine. Many of the indigenous drugs, however, after some pharmacological, chemical, and clinical investigations, were included in the British Pharmacopoeia, and some of these, like Kurki, Ispagula, Kirata, and Caladana, were accepted by the BP 1944 in Indian Pharmacopoeia list, which has been published under the authority of the government. Have these drugs been given recognition and of India, all more like Ravalthia, Pinarava, Vasik, etc., have been included. The potentiality of the Indian medicinal plants was, however, realized during the British period, and since then many workers have attempted to find out the botanical source of many of the more important drug plants through the help of Veds, Hakims, Pansaris, and local people. Chopra et al. 1956, 1958. Sir William Jones, 1799, was one of the earliest contributors in this direction. The import and contribution made hitherto in this field are John Fleming's Catalogue of Medicinal Plants 1810, Ainsley's Materia Medica of the Hindustan 1813, Roxburgh's Flora Indica 1932, Whitesicon's Plantrum Inde Orientalis 1838-58, and later the works of Royals and Essay on the Antiquity of Hindu Medicine 1837-1845, 1854. Of earlier work on Materia Medica and Pharmacology of Indian Medicinal Plants, Shaughnessy, in his book The Bengal Dispensatory and Pharmacopoeias 1844, for the first time described scientifically the properties and uses of the drug plants of Bengal. This was followed by Irvine's Materia Medica of Patna 1848, Waring's Pharmacopoeia of India 1864, Dutta's Vegetable Materia Medica of Western India 1877, but Dymock, Warden and Hooper's Pharmacographica India 1889-93, and Watt's Dictionary of Economic Produce, CTS, of India 1889-93 seem to be most valuable of all the previous works and help as an important source for scientific references. Further contributions made in this field are Day's Indigenous Drugs of India 1896, Kurtikar and Basu's Indian Medicinal Plants 1933, and Bose's Pharmacopoeia Indica 1932. In European countries and the USA, pharmacognosy is well-advanced science, and almost the entire flora has been subjected to pharmacognostic. Prologue. India, since ancient times, has a fairly well-established medical science of its own, with a rich and a veritable materia medica which includes herbal, animal, and mineral products. Although research on medical science has opened new sources of remedies, herbals are continuing as a mainstay in the treatment due to easy availability coupled with their hold on the community at large. Experts and workers on the indigenous drugs in this country and abroad have started diving deep into the ocean of this veritable herbal wealth. India, with its ecological and climatic diversities, has become a home for getting a variety of herbal material in view of widespread usage of herbs in pharmaceutical industry and therapeutics. There is a greater need for correct determination of the authentic drug and the usual adulterants, so that the genuine material can be procured in spite of having wealth of medicinal flora in India. The chief difficulty experienced by the persons dealing in the manufacture of indigenous medicine is that they find it extremely difficult to obtain genuine crude drugs. Many drugs are sold under various names, different drugs under the same name, and even in the Ayurvedic texts and materia media and books on medicinal plants, the same synonym has been given for a number of plants, creating a great confusion. The first and foremost objective, therefore, is the correct determination of the authentic drug as well as its adulterants. In order to establish the correct identity of a drug, it is essential to collect detailed information on its pharmacognostical aspects. Pharmacognosy, according to Trees 1966, Yunka 1950, and Wallace 1967, which is an applied science, aims at a complete and systematic knowledge of crude drugs of vegetable, animal, or mineral origin. It implies not only drugs, but also includes a knowledge of sources from which the drugs are prepared, their history, properties and uses, distribution, cultivation, collection, and selection. In the pursuit of this study of authenticating the plant material, a number of scientific disciplines like phytochemistry, pharmacology, etc. do have a role. Physical, sensory, botanical characteristics of drugs formed the main features of the study in the earlier period, but since the later half of the 19th century, the emphasis was laid on the crude drugs, their substitutes, adulterants, because of the commercial practices. Subsequently, when a number of drugs were introduced in the form of powders in the pharmacopoeias, a rapid progress was made in the field of microscopy. Quantitative microscopy, developed by Wallace and his associates, has proved to be of great help in the identification of drugs, which have definite countable microscopic plant structures, such as fibers, stone cells, pollen grains, trichomes, starch grains, etc.